Lecture 21.
Okay, the microphone's not working. He's coming to put new batteries in it. So I'll try and talk loud at the beginning. So today we're finishing that spinal cord lecture that we started last Tuesday, or whenever it was, seems like forever ago. And then go into lecture 19, very large lecture on brain and cranial nerves, which will extend for some time. And the iClicker questions that are at the end of the lecture today, and there will be one more Unit 3 review if you want to look at your exam after this class in room 124. Okay, so let me get started. This is a comment about when I kind of got tangled up on the lecture last time. I was talking about the tracks in the spinal cord and the ascending and descending tracks, and I gave an example, the cortical spinal ascending and descending. Oh, that's fine. Thank you. It's on mute. Hello. Thank you. So this slide just showed the descending upper motor tract, which is right here, and then lower motor tract in two different cases, one cranial and one, eventually get untangled here, and one spinal. And then I switched over and got confused. It's kind of an interesting story, but I switched over to the autonomic nervous system. I'll go through the detail at the last lecture of this unit. And the autonomic nervous system is known for its peripheral nerves, a preganglionic and postganglionic, both in the periphery. And the tracts that come down from the hypothalamus are less understood. And when I threw this up, I started thinking, yeah, I wonder where those tracks are, and all this went through my brain. And so I was kind of in my brain trying to figure out what in the world they represented, the blue lines. And so anyway, I couldn't find it in books. I talked to some of my colleagues that teach neuroscience in other medical schools or in medical schools and came out with nothing. Most people don't even refer to these tracts that innervate the autonomic nervous system. You certainly won't be tested on it, and it's seldom presented. I don't know anywhere it is. I don't think they know even where they come down the spinal cord. So anyway, when we get to the autonomic nervous system, we'll talk about pre- and post-ganglionic and what we call the thoracolumbar and the craniosacral divisions and all that. But how these are innervated to the hypothalamic area, we still are somewhat unclear on. So anyway, that's where I was stumbling around when I gave that lecture. What's said here is correct. It's just that it doesn't elaborate what these visceral motor tracts are, as it just indicates that on the diagram. Okay, so when we're starting officially today is talking about the specific photoreceptors in muscles. Very interesting and fairly complex receptor that's buried in most large muscles, large skeletal muscles of our body. They're called muscle stretch receptors, and they're essentially a bundle of specialized muscle cells that are called intrafusal fibers. The reason they're called intrafusal is to separate them. They're otherwise just muscle fibers, but to separate them from the fibers that are part of the muscle themselves that they call extrafusal. So these are a few fibers in every cell that is surrounded by a connective tissue membrane and have a set of nerves going to them that's quite complex. The distal ends of this little spindle are hooked to connective tissues that eventually couple them to the muscle themselves. So when the muscle changes its tension, either passively or actively, this also is reflected in the length of the intrafusal fibers. That turns out that there's two different classes of muscle fibers in this muscle spindle. One is referred to as nuclear bag and the other nuclear chain, because the nuclei of the one are all in a swollen area in the center, so they call it nuclear bag. Remember, all muscle cells are multinuclear. Well, this particular intrafusal fiber has its nuclei clustered at the midpoint. The other nuclear chain has the rows of nuclei all the way down the length of the intrafusal fiber. And once again, the name intrafusal separates it from the rest of the cells of that given muscle, which are referred to as extrafusal. The innervation is fairly complex. There's three different nerve families going to these muscle spindles. One are group 1A afferents, so they're sensory. They have spiral endings, and they circle both the nuclear bag and nuclear chain fibers. So those are called 1A afferent, and they're sensitive to rate of change. So again, we talk about this when we're talking about sensation of sensory receptors. That some are tonic and altered by length, absolute length, and other by rate of change. These group 1A fibers are sensitive to rate of change or rate of stretch. The group 2 afferents have spray, although this does have a spiral ending, but we usually say they have spray endings, and they are sensitive to the length of the muscle. In other words, they're just measuring the absolute length, where the others, the 1A, are measuring the change in length. And finally, there's efferent fibers also going to this intrafusal fiber. They're called gamma motor fibers, and they actually contract the muscle. These gamma fibers are just like alpha motor neurons. They're totally innervated different, but they cause contraction of these intrafusal fibers and reduce the responsiveness. And to understand that, we'll show you the next figure. So this is what would happen if you're monitoring 1A and 2 neurons coming from a spindle when you change its length. So linear stretch, you just stretch that muscle passively, you just pull it and stretch it. What will happen is the 1A that are sensitive to rate of change will send out lots of active potentials during the period of change, but once the change is over, they'll quit sending out signals. Where the 2 that are overall sensitive just to length will put out slow action potentials initially, and then faster and stay constantly fast if it's stretched. In contrast, if you tap the muscle, the group 2 fibers don't really show anything, but the tap is noted by the group 1A sensors. Sinusoidal stretch or stretch, relax, again, primarily received by 1A fibers. And a release is just the antithesis of a stretch, decrease in output from 1A during the release and decrease in rate of group 2 after the release. So anyway, we can keep track of very specifically what's going on in each muscle by information coming out of this very sensitive device. So what does the gamma motor neurons do in this situation? So this is a kind of a complex figure, but let me see if I can explain it as much as possible. So this represents an extrafusal fiber, the lower one in each case, and this represents the whole spindle. They're not trying to separate, and you don't really know much detail about it. Have to separate the two classes of intrafusal fibers, but this just represents the intrafusal fiber, and this represents a muscle spindle. So if we stretch the muscle passively with a steady stretch, you'll get a steady output of a signal from that intrafusal fiber. If the alpha motor, if you contract the extrafusal fibers, or the fibers around that muscle, it will relax the spindle inside the muscle. The muscle spindle will relax, and so the output will shut off. So when the muscle itself contracts, it would shut off the output, and this is just looking at maybe primarily group 2 afferent fibers. If you stimulate the two simultaneously, and this is where you start figuring out what gamma motor stimulation does, but if you stimulate both the interfusal and extrafusal fibers at the same time, or to put it in other words, if you stimulate the lower or the alpha motor unit and the gamma motor unit at the same time, there's not going to be much change in what the length of that spindle is. So the group 2 afferents are going to keep putting out a constant output, but the group 1 afferent is going to show the change in increase and decrease in length that occurs at the beginning and end of the stimulation. So it'll show two pulses or phasic changes as the muscle contracts and relaxes. So anyway, this gives us a really good amount of information about what's going on in the muscle during contraction. How do we use this? We use this in order to monitor or keep track of what's going on as each muscle is contracted. And I got another figure that will show that in more detail. But first I want to introduce one other proprioceptive device, and this is the Golgi tendon organ. It looks like, they called it a flower spray. It looks like a root filament or something like that at the end of the nerve that sprays out or contacts the end of every tendon, or the majority of tendons. And it's sensitive to how much stretch that tendon undergoes. The nerves that innervate it are called 1B nerves. And it's a length determiner, it's not any rate determiner. And it has a very high threshold. In other words, you really have to stretch that tendon to give any output. And when it's relaxed, it turns out it puts out kind of almost zero output. But when it's stretched, it starts putting out signals, and as it's contracted even more and more, it'll put out more signals. I think this is very confusing, but they think in most cases that the output is proportional to how much stretch. But again, it's not very sensitive, and so usually it has to be severe stretch, as you'll see in some of the figures coming up that activates. So high threshold response to stretch is probably the take-home message. So this is a kind of complex diagram, but it really is a good one because it explains what these devices are trying to do. So if you could imagine that this is the motor cortex up in the brain, and this is the area that's going to tell our muscle to contract, and over here is the muscle. So the muscle decides to contract. It sends its information to a controller, which turns out to be integrated in the cerebellum, and then that sends out alpha motor signals. Some of these don't go right to the cerebellum. Some of them just go to from the motor cortex through the alpha motor neuron to the muscle. Now, how much the muscle contracts may not be what you expect. In other words, you may be more tired than you realize, and so when you tell your muscle to contract, it doesn't follow your direction. Maybe there's a load heavier than you expect or less than you expect. So often when our motor cortex describes or contrives some motion for the muscle to activate, it doesn't respond in the way that we want it. Now, the way that we find that out is through the muscle spindle. So at the same time we tell the muscle to contract, at the same time we send a gamma motor signal to the muscle spindle and set it to about the same length. So we're going to set the interfusal fiber up contracted about the same length as the muscle spindle. And then through the interfusal fibers and their neurons, we can compare what we intended to occur to what actually occurred and send that back to the cerebellum. So we have a feedback signal from the muscle spindle that decides how much of what was intended actually occurred. In other words, we're comparing intended to actual responses. And if it doesn't respond in the way we want, we can send out another upper and lower motor, signal back to the muscle that we're trying to move to make it more accurate. So it's called the servo control mechanism. In other words, we're always comparing what we intend to what actually occurs, and the way which we do that is by the muscle spindle. So it's kind of our reference base. We get its reference by telling it what we wanted by contracting the gamma motor neuron, by contracting the muscle coupled to the gamma motor neuron, and then compare that to the extrafusal fiber. So it's kind of an interesting system, and it's certainly amazing how complex we've evolved it. So anyway, that's a little bit about proprioceptors. We'll talk about somewhat more when we get to reflexes, as we're going right now, because some of them are involved pretty heavily in our reflex system. The reflexes are usually unconscious, but they're fast, predictable, automatic responses to environmental change. That's probably the most succinct definition we can come up with. They can be classified by the sensory and effector limbs of the reflex into categories. For example, a visceral reflex is one that both arms, both the sensory and effector parts, are in the viscera. And the baroreceptors are an example of that. Baroreceptors, and we'll talk about them in detail in the second semester, are sensitive to our blood pressure, and when it goes up or down, they cause increases in heart rate, and stroke volume, and changes in peripheral resistance, and all kinds of integrated responses. We're not even aware of it. So the sensor is visceral, it's part of our vasculature, and the response is in the heart, and blood vessels. So none of these are conscious, so it's a sensory effector, or both visceral. A viscerosomatic has a somatic sensory component and a visceral sensory component and a somatic effector. So the sensory is always the first one of the contractual terms. So the appendix rupture is the visceral sensory thing. Stretch receptors and pain receptors in the appendix are activated if it's stretched so much that it ruptures, and that causes a contraction of the somatic muscles in the abdomen, like you experience with cramping or pain. So that would be a visceral somatic kind of reflex. Somatovisceral starts out in the soma, in this case receptors that sense temperature in your skin and causes a visceral response that we're probably not aware of. So when we're cool, our blood vessels vasoconstrict, so we shunt the blood flow to the core of the body so we don't lose that temperature. So that's originates for receptors in the soma, we call it, and then the visceral respond. And finally, most of the reflexes that we are conscious of, that we're aware of, are somatosomatic, and the sensory and effectors are both in the somatic nervous system. And there we have a great example is the one we're going to talk about in a second, the knee tap reflex, where we stretch a muscle and the muscle, the ipsilateral muscle, contracts in response to that. Another classification of reflexes is whether they're monosynaptic or polysynaptic. Monosynaptic reflex has a sensory neuron, an afferent neuron that goes up to the spinal cord and directly synapses with the effector or efferent neuron that goes back to the muscle usually. So it's a single two-neuron arc, and we call it monosynaptic. A polysynaptic has usually a interneuron or more, so the information is integrated a little bit more. A withdrawal reflex, it involves several different muscle families, the classic polysynaptic, where you hurt yourself, you burn your hand, and you withdraw it. That would involve activity of several interneurons. The other thing, general comments about different reflexes before we start talking to them specifically are classic elements that all of them have in common. For example, you have to begin with some receptor, and those receptors the modalities that they're activated by can vary from pain to stretch to temperature to every type of sensation practically that receptors are activated for. So a variety of stimuli are activated to initiate these responses. These receptors are often coupled to or themselves sensory neurons that conduct that information back to the through the dorsal root to the nerve cord. And then within the gray matter, within the butterfly cord, then that's the integrating center. If it's a monosynaptic reflex, it just goes right into the afferent neuron. If it's polysynaptic, it couples with some interneuron in the gray matter of the cord. And finally, a motor afferent neuron goes through the ventral root and out to the effector. So those are the four classic components: receptor, sensory neuron, integrating center, and motor neuron. And the motor neuron often couples to, and most often couples to the muscle as the effector. So the first one we'll talk about are it's called phasic reflexes, and the knee-jerk reflex is an example of it. You've all had this done. The physician taps the patellar ligament, and that stretches the muscle that has within it a muscle spindle. So the receptor is a muscle spindle and stretches a muscle spindle within the muscle, and that sends a signal largely through from 1A afferents to the posterior horn of the cord, and goes up. And this does not have an interneuron. It's a classic monosynaptic reflex. Goes back the ipsilateral side out the ventral horn or anterior horn back to the same muscle. And so the physician taps the tendon, and you kick your foot out and hopefully hit him, so he's punished for fooling with you. What's interesting is, what's the value? Why did we develop this? So physicians can play with us. I mean, why did we come up with this, evolve this reflex? Well, if you're trying to think of a value for it, one value is if let's say you're going down a stairway, let's make it interesting, and you're with a colleague talking as you go and not paying attention, and you thought you were on the landing, but there was one more step there. And so you unexpectedly stretch your muscle more than you would, and that unexpected stretch causes that same muscle to contract. So instead of collapsing when you hit the landing, you're cool. You just kind of like that, and your colleague doesn't know what happened. So anyway, it helps us maintain balance when our muscles stretch in unusual ways. Many years ago, a man developed a maneuver that reinforces this, and I don't know if you can see this from where you're sitting, but he taps the tendon, and he has the patient grip his two arms together and pull apart, like clasping his palms. So he pulls apart his arms and puts effort into that. You spend a lot of time explaining. Now, what I want you to do is pull your arms apart, and the patient is thinking about all this and making sure he does it right. And then you tap his tendon, and his knee goes out about twice as far. And the reason is that we're always have the ability very easily to inhibit things, and when we're thinking about the physician tapping your patellar tendon, you're thinking, I'm going to kick out or something, and for some reason you tend to inhibit it. But when you're concentrating on your arms, that inhibition is decreased, and it always ends up with a more amazing, stronger reflex. We have, as I said, a lot of tonic activity all the time to our muscles that gives it, instead of just being totally relaxed and with our muscles, there's always some tone. And if a physician takes your arm there will always be resistance, even though it's fairly minor, unless you're in a coma or something, to its movement. And so slow movement causes some resistance to that movement, and it's induced also by both 1A and 2A fibers from muscle spindles, and it just maintains some slight stretch of the same muscles being stretched. So it's maintaining us in position, essentially, they think, all the time, so that we're just not completely relaxed. The tendon reflex is that one that I talked about, that the receptor is that flower spray device in the tendon, and it's really there just to protect the muscle that it's coupled with, so that the tendon doesn't tear. So it's not activated unless there's a tremendous amount of force in the tendon, and then it inhibits that same muscle from activating anymore, so it prevents, hopefully, the tendon being torn. You'll Saturday Night Live episode of Hans and Franz doing these exercises probably decades ago, but maybe you've seen some of the replays of it. But one episode they were showing off their curls, and they ripped a muscle right off the arm, and it went curling down. That actually happens in gyms, I'm told, where some of these guys work out so much. With continued working out, you can inhibit this Golgi tendon organ, and it doesn't work as well as it should. But the idea is that if you put too much force, it'll inhibit the muscle contracting, and you won't be able to tear the tendon. Okay, these are polysynaptic reflexes, and they're more complex in that they go into the spinal cord and require either, it's called an interneuron, an association neuron that couples to often more than one, or even decussates and couples to neurons on the other side, or contralateral side. The first part of this is a withdrawal reflex, and then we'll talk about the crossed extensor reflex. But this is the thing I think we think of mostly when we think of reflexes. If you if your hand touches something hot, or if you cut yourself with a pen or a knife or something, you, by reflex, move that limb from the injury site without even any thought, and that's a classic reflex. So it's activated by pain receptors. The impulse goes up into the, initially by C fibers, up into the spinal cord, and activates association neurons, and you have to pull flexor muscles that usually involve several different muscles, that withdraws the limb from the injury. So that's the classic withdrawal reflex. The same time that's going on, there's a crossed extensor reflex that's involved with the association neuron. So while you're withdrawing your arm, they've got it over on this side now, while you're withdrawing your arm due to pain in that limb, it causes the opposite arm to go, if you're withdrawing, the opposite arm will extend, and it's to maintain posture. So let's say you burn your hand, you'll pull your hand away from the site of injury and extend the other one, and that keeps your balance. So it's a crossed extensor reflex. It also occurs, when you step on something sharp, you withdraw that limb and you extend the other so you don't end up on the floor. So it does have a lot of basis. I don't know if you ever, I remember the exact incident, if you've been by somebody when they've injured themselves. Like one time I was standing by this person, they burned their hand on the stove, and I got slugged. So often the person right beside that gets the effect of the crossed extensor response. Some of these, there's always some inhibition going on in all these pathways, and it's part of maintaining some tone. But as we age, like everything else, some of this tone disappears. And I don't know if you've had, myself, some aged professors, but some of these reflexes appear even during, inappropriate, I guess you could say, time. For example, I remember a professor that did a lot of writing on the blackboard, and every time he moved his one arm up, the other one went down. And so this is, some of that cross-extension activity happening just because these reflexes kind of lose their inhibition as we age. What's the value of evaluating reflexes in patients? Well, a simple one is that's how we tell the level of anesthesia. If you're putting a patient under anesthesia, one of the classic things is to touch their eyelid and see it, or their eyes, if their eyelid flashes, pinch their finger and see if they withdraw. So you can easily tell how deep they are in anesthesia. In fact, there's levels of tests as you go deeper and deeper into anesthesia. The patients that have intervertebral discs often have sometimes loss and sometimes increase in, depends on the type of injury on reflexes, and finally all kinds of muscular weakness diseases. Patients lose the amplitude of reflexes due to the loss of function. For example, myasthenia gravis, where you lose the neuromuscular receptors. Wanted to talk a little bit about spinal cord shock and what occurs with unfortunate transections of the spinal cord. Initially, after there's a transection or loss of conduction to the spinal cord, there's no reflexes below there, and they think it's just complete loss of, there's always inhibitory traffic, but there's always excitatory traffic going down the spinal cord. So both tone is maintained by a little bit of activity from both sides of the system. If you lose the excitatory output, you can't get any reflexes, and so initially you test the patient and you won't see any reflexes at all. After recovery, day 1 through 3, you'll start getting stretch and flexion reflexes, and first polysynaptic evidently, and then later monosynaptic, and they're more exaggerated than they normally are. And again, that's because we have inhibitory traffic that comes down to those things. That's why we do the gendrastic maneuver. You have some conscious or subconscious inhibitory activity on muscles all the time. So when you lose that coming through below that transection, then you have exaggerated reflexes. And then weeks later, these reflexes even get larger with very little stimulus. You get huge responses, and they think some of the neurons actually sprout new dendrites, and you start having new connections that form below the injury site. So these are classic changes that occur following spinal cord transection. You might have heard about Babinski's sign. If you tickle a small child's base of his foot, his toes curl up or fan up. Like the most of us, the contraction is downward. So this is a normal plantar reflex. In an infant, the first few weeks, he does the opposite. It's called Babinski's sign. But if you damage the upper motor neuron, you'll get the same response. So whatever is causing that to occur, this Babinski's sign occurs with corticospinal damage, and we don't know exactly why, but it's one way to find out if that tract has been damaged. If you transect much higher than the cord up in the brainstem, you'll have exaggerated extensor activity such that the person it's called decerebrate rigidity, and it's due to these stretch reflexes that become ridiculously hyperactive, and the only way to stop this person from being in the seizure condition is to cut dorsal roots to denervate these muscle groups. And finally, a little bit about postural reflexes. We've mentioned some of the eye briefly before, but these again depend on proprioceptors, but when we're talking about posture, usually they're vestibular apparatus or cranial propioreceptors. And the first ones are those that regulate our eye movement, and it usually comes from the devices in the, well, we talked about the inner ear last week or so. The first and simplest is when you look at something and turn your head, you maintain focus on that object by a reflex that senses the motion of your head, and lets your eyes and moves your eye in the opposite way, so you can maintain that object in focus. If you continue to turn the person, you get what's called nystagmus, and you can do this on an individual. You can put him on some chair like this chair here and spin them around and look at their eyes, and they'll focus on an object, and so their eyes will go the opposite way their head does, and then they'll come back fast to find another object, and they'll go like that. So their eyes go fast in the direction the head is turning, and then slow, focusing on an object in the opposite direction. So they have these alternating fast and slow movements, and that's called nystagmus. We all do that when they're put in a spinning situation. Vestibular placing reaction, that's you can test this if you have a cat. You throw the cat up in the air, and what happens is they go like this, and so when they land, or if you're strong, you could throw your special other person in the air. And anyway, when they land, we all put our arms out and our limbs out in order to make the landing softer. We also have some oscillating circuits that make it easier to walk and to do constant activities. And the best one that we can understand is our walking. Pressure on our foot pad causes an alternate efferent activity. And so when you walk, you just start a circuit that turns a set of muscles on alternately in each side of our body. And you can even mimic this on a cat that's got a spinal cord transection. If you place them on a walking treadmill, it'll walk normally. So a lot of our walking activity doesn't require central nervous activity. It's a spinal cord reflex. Okay, so that's it. We're not going to do eye flickers yet. I'm going to get a little bit into the next lecture just to not get too far behind for about 5 or 10 minutes. The next lecture is on the brain, and we'll end it up with a discussion on the cranial nerve. But what I'm going to do is go through the different parts of the brain and discuss generally what each is thought to do, and mostly anatomically their structure. And then after that we have a lecture on higher functions, memory and sleep and things like that. So first, a little bit about anatomically about the brain. The brain also is responsible for reflexes, just like we talked about spinal cord reflexes, which we just did. There are reflexes that require the brain's activity. Those visual reflexes do, but we also have respiratory and cardiovascular reflexes that are coordinated in the central nervous system and the cranium. So it's also a reflex center. There's a lot of subconscious activity that occurs in the brain. We'll talk about that. And then, of course, that's the center where we store information and memory. We have conscious activity and even abstract thought. The brain starts out during embryological development as 3 swellings in the cephalic portion of the spinal cord in even primitive worms. So even in the simplest of organisms, during their development, there's 3 swollen portions at the cephalic end of the spinal cord. And we've referred to them as the prosencephalon, the mesencephalon, the rhombencephalon. In humans, they develop into the parts of the brain. So the prosencephalon develops a little further along by the 6th week or so of development into 2 portions. The telencephalon and the diencephalon become the cerebrum, the cerebral hemispheres, and the diencephalon. We'll talk about that later. And then the midbrain stays the midbrain, mesencephalon, and the rhombencephalon divides into the metencephalon and the myelencephalon that eventually become the cerebellum and the pons, and the myelencephalon becomes the medulla oblongata. And we're going to talk about all these little parts as we go through the lectures and what each one's responsible for. The adult brain has an area which we call the brain stem, which, well, first of all, they said, and I don't know who could have counted these. I don't even know what 100 billion neurons means, so I said, all I know is it's more than 6, but I have no idea what that means. But somebody estimates there's that many different nerve cells. The major components are the brainstem, which is largely hidden here under the huge cerebral hemispheres and behind the cerebellum. And from the brainstem, we go into the diencephalon. The cerebrum is the large cerebral hemisphere area, and then there's other large spherical structures, of course, the cerebellum. Those are principal components of the human brain. So we're going to start at the most inferior and move up and talk a little bit about each one. The most inferior part of the brainstem is called the medulla oblongata. And as you could imagine, all traffic going up to the brain and down to the spinal cord has to go through that area. So we say all ascending and descending tracks go through there. Most nerves decussate across in the medulla, but as we said, in the pain situation, others, sometimes the decussation occurs in the spinal cord. There are several different control centers in the medulla, and we'll hear more about these second semester. There's a cardiac center that controls heart activity, and a cardiovascular center controls vasomotion. A respiratory center is vomiting, coughing, and swallowing. These are all parts or little medulla or areas within or nuclei within the medulla. Just above the medulla, which is here in red, there's a swollen area a little bit larger in diameter called the pons, and that term means bridge. And it's swollen on the inferior side of the brainstem, and it's opposite to the cerebellum. And what the large girth is, because nerve tracks wrap around this area, go over to the cerebellum. So we call these peduncles, but they're essentially like wired bundles, like you were wiring some very complex electronic device that come out of the medulla and wrap around and go into the cerebellum. So these large tracks, of which there's 3, and I'll talk about it more in a second, are called cerebellar peduncles. They're going from the pons into the cerebellum. Also in the pons are some control centers, just like in the medulla. Most of these are respiratory. The apnoistic and pneumotactic center are both respiratory control centers that control our rate of breathing, and I'll save their function until we get to the respiratory system. And then on the back, this is a superior view, obviously. On the back of looking from the dorsal side, you can see the cerebellum right inferior to the cerebrum, and it's classically called the second largest structure of the cranial cavity. And here's a magnification photo, and you'll see it's divided into 2 hemispheres, just like the cerebrum is, and it's joined by a narrow lobular structure, which is called the vermis. So there's 2 lobes of the cerebellum, and they're joined by this structure in the middle called the vermis. If you cut across the cerebellum, you'll see this tree-like structure, and it's white tracks going up to the surface that has gray matter. So there's a gray matter cortex of the cerebellum, just like there is of the cerebrum, we'll talk about later, and the tracks going up there form white branches. Sometimes they call this arbor vitae, which stands for tree of life. So it looks like a tree with leaves coming out, but the arbor vitae, the arbor, the branches are the tracks going up to the cell bodies out on the surface, just like in the cerebrum. And then I mentioned those cerebral peduncles that form the pons. There's a superior, an inferior, and a middle. So once again, these are tracks going to the cerebellum. The superior tract has fibers coming from the motor cortex to the cerebellum. So when we want our muscles to move, we tell our cerebellum what our intention is, as well as the muscles, so the cerebellum can coordinate that activity. The middle tract contains fibers from the motor cortex back to the, I said that backwards. The superior is cerebellum to the motor cortex. The middle is motor cortex to the cerebellum. So there's this communication going on between the 2, and the way what they're trying to do is integrate, as we said before, the vestibular and proprioceptor activity. So the inferior tract right here has information coming from the spindles and from the Golgi apparatus and all those joint receptors and so forth, coming up and going into the cerebellum. So the cerebellum receives information from the motor cortex on what's wanted, information from the muscle on what's going on, and then it makes, minute changes and send that up to the motor cortex to change what's going on. So that's the integrating center of our muscle activity. Controlled coordination of muscle activity compares intended for actual. When you have cerebellar deficits or lose cerebellar activity, you have gross problems in muscular activity. Some of it's just tremors. Sometimes you miss things. They call it past reaching, where you try to grab something but you'll miss it. I hate when that happens. So you just can't quite do what you want to do. You lose some of the fine motor control. Okay, so that's, I think, where I wanted to get to today. And next time we'll start from here, where we go from the cerebellum up through the midbrain and so forth. So this is, I figured I'd get. So we'll do some eye clickers if you have your eye clickers with you. No one asked me. I'm kind of surprised I didn't get the email like I usually do. Are we going to be tested on the pain lecture? Yeah, I will have a couple of questions. They won't be little detailed questions, but this is some examples of a question that, in fact, I had her do these. Some simple questions on pain that are from that lecture. So these are from the pain lecture last time. Okay, so the first one. Which of the following is not correct regarding pain sensation? Okay, 5 seconds. Okay. What we wanted was C, which is it's conducted by C fibers. Slow. What? Which one? We wanted this one. Yeah, it's conducted by C fibers and not by A delta fibers. Oh, okay. Yeah, maybe you're right. Maybe there's I'm not a pain person. But I guess D is also correct because acute pain or sharp pain is exactly localized, where the second pain is usually much more diffuse. They're all correct. Is not correct. Which is not correct? They're all correct. Well, what I thought was not correct was C. So anyway, let me talk to her, see if she will agree. Okay, allodynia. That one's pretty fine. Looks like everybody agrees. Okay, 5 seconds. Yep, that's what we were looking for its innocuous stimuli cause pain that which shouldn't, in other words. Okay, number 3. Which of the following is correct concerning the transmission of pain within the central nervous system? Five seconds. Okay, looks like we'll, they certainly don't decussate the medulla, they decussate immediately. Pain traffic does go up the spinal thalamic tract. The cell body of the second, I think it's the third, I'll look that up. That's what most of you chose. I think it decussates and immediately sends another one up, but I may be wrong. So maybe 2 are right. I'm going to be good. You can tell I'm not a pain expert. And they don't share the same tract as proprioception, so it's B or C. How am I doing? This one I know is correct. Okay, let's try this. Ten seconds. Okay. Yeah, this sodium never has been affiliated with it. It's first of all not found hardly at all intracellularly, but it's never been known to be a pain mediator. So A, and number 5. So, neuropathic pain, you're right, is when the tissue itself is damaged, either central or peripheral, so E is correct. Okay, I'm going to go to room 124 if you can get your exam.